So the next part of this symposium, which I'm very pleased to be part of, is on featuring some of the MCHRI faculty scholars. Faculty scholars have been supported by a number of our donors. It was mentioned both by Mary and Cynthia, and we're indebted to them. And these scholars represent both pediatric investigators or investigators who are also doing work that can affect the life and health of pregnant women and children. And today we're going to have two speakers, neither of which are in the Department of Pediatrics, but both of whom have really done important work on helping us understand what happens in the health of pregnant women and children. The first speaker is Dr. Catherine Blish, who is an associate professor of medicine and is the Tasha and John Morgridge faculty scholar in pediatric translational medicine. Now, I'm not allowed to say a lot about her. I was told not to go through her biography and the like, but I do want to say that her laboratory is involved with developing new methods to prevent and control infectious diseases through better understanding of human immunology. Most of her work has been on NK cells, but she also has a really important interest on the immune signatures of human pregnancy. So today she's going to speak with us about why are pregnant women more susceptible to complications from influenza. Dr. Blish. And she's amazing. She's limping on her way over here, but undaunted, she's going to give us her talk. Thanks.
I'll just say now, thank you very much. It's really an honor to be here and have a chance to talk about some of the work that my lab has done, complete with my newest accessory here. So hold on, give me a second here. Largely through the support of the Maternal and Child Health Research Institute. And this has really been exciting because, as Harvey alluded to, I'm an adult ID doc. And therefore, this sort of realm into maternal fetal medicine has been an interesting passion, but not necessarily the type of thing that was easy to get NIH funding for in the initial stages. But I wanted to share just a little vignette about the sort of obsession in my laboratory about why pregnant women are so susceptible to influenza. And just to begin, I wanted to put this in context as to why I became interested in this question. And if you look at the data, back in the 1918 pandemic, the mortality rate among the general population who became infected with the 1918 pandemic strain was 2 to 3%. But it was an astounding 27 to 75% among pregnant women that were infected at that time. I mean, think about that. Almost half or so, the estimates are wrong, of the pregnant women who happened to get that flu strain died. Now, of course, we have much better preventive care now and much better supportive care for individuals who become flu infected. But even in the 2009 pandemic, where the mortality rate was 0.9 per 1,000 cases among the general population, or sorry, 0.4, it was still 0.9, more than double among pregnant women who are generally young and fairly healthy. So, and the question is, does this occur just in pandemics? It's a little unclear, but even with seasonal influenza, there are much greater rates of hospitalization and ICU admission among pregnant women. Now, as somebody who's had 2 children myself, I always wonder if we just are more likely to hospitalize pregnant women because we worry about them. But I still think it's hard to argue with this mortality. And as somebody whose lab focuses on immunology, of course, one of my major questions is how does the immune system mediate this susceptibility and what are the changes? And this is just a figure from a nice paper by Catherine Curtis. Oh, interesting. I hope this doesn't occur throughout. My animations aren't working properly. But at any rate. So this is a nice paper from Catherine Curtis explaining that as the progesterone and estradiol levels go up during pregnancy, there's not just flu, but a wide variety of infections to which pregnant women are more susceptible. And this is thought to be associated with an increase in the function of our innate immune system, primarily, with our monocytes, dendritic cells, et cetera, going up in function, but a decline in our adaptive immune system to prevent rejection of this foreign fetus. Now, we do a lot of work in our lab looking not just at NK cells in this, but I'm going to focus on natural killer cells in this story for the sake of time, primarily because it is a major focus of my laboratory, but also because they're a little curious. They're actually innate cells, but they're classified in the adaptive in this response, in the thought that because of their ability to respond very rapidly to viral infection or to tumor cells, that they must be downregulated during pregnancy in order to prevent rejection of the fetus. And just to give a little bit more background on why we're interested in natural killer cells in this situation, I'm going to start with just a little picture as to why we'd even care about these cells. The blue cell there is a natural killer cell. It's got a surface stain. It's been dyed with a cytoplasmic dye. And it's sitting next to a monocyte from the same person. And you can see that it's just this boring round cell. Why would we care? It doesn't have the beautiful antigen-specific receptor that a T cell would have. It doesn't make antibodies. And yet, if we take these cells from the same individual, this is the same NK cell and a monocyte from the same individual. But the only difference is we infected that monocyte with flu. And now you can see that the NK cell is no longer this boring round cell. In fact, it's got this ruffled membrane, it's polarized, and it's accumulated a bunch of material right at that intersection with that infected cell. So it's somehow sensed that the cell is infected, it's getting ready to kill it, and it's formed this immune synapse, this accumulation that allows it to talk to this cell and potentially kill it, which is likely what's going to be the outcome of this. And so one of the questions is, how does this happen? And just to see this in action, the green cell here is an NK cell, and the red cell is a tumor cell. And you can see this NK cell pushing and prodding and forming that immune synapse, and the cell swells, and when that blue dye appeared, that tumor cell was dead. And so one of the questions is, you've got this very potent antiviral activity. The ability to do this, as you can see, in the course of minutes, and this NK cell can disengage and go on and do this to the next cell very quickly. And so you can see that you wouldn't necessarily want these cells to be cruising around right at the maternal-fetal interface, rejecting the foreign baby cells. So the question is, what is happening to their function, and could this play a role, since they're also important for fighting viruses, in the susceptibility to flu? And now, I told you, and this is a very silly cartoon, but I hope we'll get the idea across, that NK cells kill, and in fact, NK cells can kill. And that is the first action that I'm going to show you here in a little claymation that a former grad student made. So direct cytolysis is what I just showed you, where an NK cell can go, and it can inject perforin, which punches holes in the cell, and then the granzymes go in and resolve that cell from the inside out. But that's not the only thing NK cells do. They can also kill through ligating death receptors like fast ligand, trail TNFR, also through antibody-dependent cellular cytotoxicity. So it's a whole new method to kill a cell. But very importantly, NK cells also are major secretors of cytokines, and they're very early responders, and they can recruit other cells to sites of inflammation. So as you can see in this case, the NK cell is sending a signal to the T cell in this case, who's doing its dirty work, and it's quite pleased with the effect. So with this in mind, our big question was, which of these functions could be different in pregnancy, and how is this accounting for this balance between the ability to fight flu and the ability to respond to an infection, to tolerate the fetus? So obviously I'm trying to keep this a little light because rather than going strongly data heavy. So several years ago when I started my lab, working with Alex Kay, who was a pediatric ID fellow who was subsequently supported actually also by CHRI, we wanted to really ask what defects in NK cell function required to tolerate the fetus played a role in influenza susceptibility. And we took advantage of an ongoing study here at Stanford that was looking at flu vaccine, because it's the one time you can kind of mess with humans on purpose and know exactly what you did and when you did it. And the only problem with the ongoing studies was they were excluding pregnant women, which we thought was the most important group, in fact, to study. So we got some funding and we enrolled a little pilot study to look at pregnant compared to non-pregnant women receiving flu vaccine. And we collected their blood before and after vaccination. And in the pregnant women, we also collected a postpartum sample so we could see how their immune system was different before and after, although obviously a lot has happened in between in terms of vaccination and exposure. And when Alex then looked at the natural killer cells, he first stimulated them with p-aminomycin, this non-specific stimulus that really is a sledgehammer for NK cells. And what he saw was that indeed the NK cells were suppressed in their ability to induce interferon gamma, a major pro-inflammatory cytokine. So the pregnant cells are in the orange, the blue dots are the control individuals before and after vaccination. So you can see that. And the same was seen among T cells, as you can see. The reality is, though, we thought, unlike most of the prior studies, that if we were interested in flu, we should actually look at flu-specific responses. And this is where things got a little different. When we actually looked at the ability of NK cells to respond to flu-infected cells, we observed that the NK cells were not suppressed. They actually had enhanced function. And this was true a little bit for interferon gamma, mostly post-vaccination, which is intriguing in of itself, but also for MIP-1 beta, which is one of those chemokines that attracts the other cells to the site of inflammation. And CD107A is an indirect marker of that killing activity. So all of these functions were actually higher in pregnancy than not. Now, I'm not going to go into too much detail. This work was published a few years ago, but it laid out several important factors that we've actually spent the last 4 years trying to dissect out. The first is the fact that the immunodeficiency of pregnancy is a massive overgeneralization. And in fact, it really depends on what the stimulus is and what the context is and what kind of immune response you're going to see and where. But the other question is really, is it maybe an enhanced, not a diminished NK cell response that's contributing to flu pathogenesis? And in fact, the only person who wasn't terribly surprised by this data was Natalie Aziz, our OB collaborator, who's like, of course it's a pro-inflammatory condition. So with that in mind, we still wanted to know what was going on. And we also caught a fair bit of flack for this data because frankly, nobody believed it. We all know that most autoimmune diseases get better during pregnancy, not worse, but at most, not all, and they weren't looking at flu responses. So subsequently, a new postdoc, Alex had moved on to a faculty position by this point, but new postdoc took a second cohort of pregnant women and decided to look if there was really an intrinsic NK cell difference, then NK cells from pregnant women should respond better to tumor targets. And so he did the simple experiment. He sorted NK cells from pregnant versus non-pregnant women. The pregnant women are in red, and you can see a better ability to induce this killing molecule on NK cells, and in fact, more tumor cells killed. So this really indicates that there is certain cytolytic functions that are enhanced, not suppressed, during pregnancy. And he also looked at flu responses and confirmed Alex's earlier results. But the question was really, why was this the case? And in parallel, we were undertaking a very broad systems evaluation of flu responses during pregnancy that wasn't just focused on NK cells, but also antibody responses, T cells, and everything else. But again, I'm just going to talk about the NK cells. And like all systems responses, there could be about 10 more people on this slide, but this was work started by Alex and Nick, who was a grad student in the lab, completed by Mathieu, and very critically, our statistical collaborators, Susan and Christophe. So basically, we took 2 different cohorts of pregnant and non-pregnant women. We looked at their blood. We looked by cytoph at a whole range of molecules. And we developed with Susan a generalized linear mix models to identify which markers were significantly predictive of pregnancy or not. And when we did this, we found that, surprisingly, there was the top hit in our first cohort. In this case, I'm just showing the pregnant versus postpartum comparison, but it was the same cross-sectionally in pregnant versus non-pregnant women. That the top hit, most predictive of pregnancy, so anything on the right is predictive of pregnancy, anything on the left predictive of not being pregnant, if we look just at NK cells, was CD38, which is thought of generally as an activation marker on T cells, but in fact is more highly expressed on NK cells. But nobody cares about NK cells except us, I think. And in fact, if we'd thought to even look at this, we didn't need to do cytoph at all. We could have just stained by flow, because if you look, it's clearly higher in pregnant women, coming down to normal in postpartum by 6 weeks, and a high distribution in non-pregnant women. And the second cohort completely reaffirmed these results. It was the top hit in both cohorts of women receiving different flu vaccines. So the question is, what is CD38 and what does this mean? And just as a very brief introduction, we weren't expecting to see this, CD38 actually is best known as not just an activation marker, but an extracellular ectoenzyme that breaks down NAD into either cyclic ADPR or NADPH, both of which can diffuse across the membrane and activate a cell. And this has been studied in T cells and a whole range of cells. And in fact, there are 8 million therapeutics on the market using CD38 to treat everything from obesity to early aging, based on this enzymatic activity and its effect in the nervous system. But intriguingly, there's a less well-known activity of CD38, which is that it can bind CD31, which is expressed ubiquitously throughout the body, but particularly on endothelial cells, but also on most immune cell subsets. And it's actually thought to play a role in adhesion and extravasation of lymphocytes into sites of inflammation. So we wondered, do either of these activities play any role, or is CD38 just marking a more functional subset of NK cells? And so Mathieu first just sorted CD38 positive and negative cells and showed directly that CD38 positive NK cells are far more functional in their ability to generate killing activity, to secrete interferon gamma, the pro-inflammatory cytokine, and to actually kill infected cells. So clearly there's something. And we did in fact find that CD38 marks a much more mature phenotype of NK cells. But importantly and critically, blocking CD38 with a blocking antibody in the red bars significantly abrogated the NK cell response. And so this is really the first demonstration that CD38 actually does anything on NK cells, and much of anything on T cells for that matter, if this were to hold in T cells. And so this occurred whether we looked at that killing activity, the cytokine secretion, or just the number of cells killed. So it's clearly important for this intrinsic NK cell function. Now, of course, unfortunately, before we did this, we spent about a year playing around with various enzymatic activities because that was the best known function, and none of them had any effect whatsoever on NK cell function. But the adhesion was interesting. And given that the potential of this adhesion-like molecule, Mathieu wondered whether this could contribute to that formation of that immune synapse. And in fact, I showed you these images before. I just didn't tell you what you were looking at. These are the same NK cells and the same monocyte. And just as an example, but in the top panels, we didn't infect the monocytes. And you can see that the NK cell, which is stained the red dye, is CD38. It's expressed on both the monocytes and the NK cells. But you don't see accumulation. However, once you infect the monocytes, you see this massive accumulation of CD38 co-localizing with LFA1, which is a canonical marker of that immune synapse, right at that site of interaction. And in fact, blocking the ability of CD38 to bind to CD31 inhibits synapse formation, and the cells could no longer kill. And you couldn't see any synapses. And this worked not just for flu, but for tumors, which is very, very interesting. So really, to bring this to a conclusion, what I hope I've told you a little story about is really, I think of the balance of immunity during pregnancy as a bit of a Goldilocks phenomenon. And in particular, when we think about NK cell activity upon influenza infection, that really what we're looking for is that beautiful green zone in the middle, where you have just enough NK cell activity to clear the virus and limited lung injury, and you can do pretty well. Whereas if you have too little NK cell activity, and this is partly based on the mouse model, the virus can replicate out of control and cause direct damage to the lung. But unfortunately, too much NK cell activity can be equally counterproductive. You might clear the virus, but you're going to cause inflammatory lung injury. And I would argue that pregnant women are on that side of this equation. And in fact, one of the key molecules that's driving this, surprisingly, is CD38, for which there are already a number of therapeutics, intriguingly, already in motion for things like multiple myeloma, et cetera. And I would also like to make the point that this is sort of an interesting example of how a systems approach, which I think can be really kind of creamed at study section and so forth for being too descriptive, can actually lead to new biology in places that were perhaps right in front of you, but you never would have thought to look at it. The only reason CD38 was even in this panel was because we were looking at plasmablast responses on B cells. And we never would have thought to look at its function on natural killer cells. So with that, I just want to thank everyone in my lab. As you can see, this was primarily the work of Mathieu Lagarze over the last several years, but begun by Alex and Nick and my outstanding lab manager, Tanmay. I'm not sure why it is that the men in my lab all study pregnancy and the women do everything else, but we can have that discussion later. And I'm really grateful for all my collaborators and the incredible support of the CHRI. So thank you.
Okay. Thank you. Questions for Catherine? So, Catherine, I got a question about flu in general and whether this relates to the severity of flu in general. And is it those individuals who have more NK cell activity or CD38 are going to have a more severe flu?
That's a really good question. We're trying to figure that out. So we do see that during acute flu in healthy individuals, CD38 is induced as part of the normal response. But what we don't have is a large enough cohort at this point to say whether those who were lower before do more poorly. But intriguingly, pregnant women at baseline look like a non-pregnant person with flu. So it's like almost the exact same level. So pregnant women look like they already have flu based on their CD38 expression. And so my theory is that that is basically screwed up homeostasis, and that's why they can't deal with the added insult of that further increase and have trouble coming back to homeostasis. But we are enrolling, but we don't have enough patients to make any valid conclusions. That's based off of six non-pregnant and 6 pregnant people infected with flu, where we've got acute and convalescent.
Great. Other questions?
Yeah.
Yes.
Thank you. Okay, so the first question was, did we look at treatment modalities? And the short answer to that is no. As I alluded to, most of the data I showed was in pregnant women who weren't actually infected. We were taking their cells and infecting them in vitro. and I perhaps should have mentioned that, to mimic infection. And we were vaccinating them and looking at their vaccine responses. We've only got a very small cohort of pregnant women with actual flu who we have collected samples from, and it's not enough that we can look at what different antivirals they receive yet and distinguish those. The second question, I'm not sure I entirely followed. Absolutely. Yeah, no, I mean, that's a very good point. It would be very interesting if we had enrolled these women for continued follow-up, since we have what they looked like during pregnancy. We do have a much smaller study that we haven't looked at yet, where we were actually collecting blood from women who were trying to conceive, and so we actually have their pre-pregnancy and their during pregnancy samples, but needless to say, that was a rather ambitious attempt to enroll, and enrollment is very poor and follow through.
Thanks so much.
Thank you.
Can I give you a hand?